ourselves. It's Brooks' quote, and I'm unquote now. It's a very beautiful quote I received by Dr. Sumaya Madam when she sent the invitation letters to me. Friends, if you just look at 1 year before only, then we never could have imagined that seminars or such webinars will be conducted virtually or online. But the pandemic COVID-19 gave us this opportunity. So, very good morning, ladies and gentlemen. I'm super excited to have you here. Before begin my speech, I must thanks to Principal Dr. Magdum Faruqi sir, Dr. Rafiq Zakaria College for the Women. I'm especially Dr. Sumaya Fatima Madam, Professor Sumaya Fatima Madam, Head Department of Botany, Dr. Rafiq Zakaria College for Women, for inviting me for this today's session. So, friends, today I selected the topic that is current developments in the mycorrhizal fungi for this today's webinar. It is very interesting relationship between fungi and higher plants, especially the vascular plants. It's a symbiotic relationship between these two different organisms from different groups. One is completely heterotrophs and another, they are the autotrophs. It was during the last decades of the 19th century that VA mycorrhizal fungi was discovered and described. Actually, it's a widespread interaction between the different plants, groups, as well as from the fungi. They were actually reviewed by Reiner in 1927 and later on by Goodman in 1955. It will be very interesting to know that more than 95% of the entire plants present on these planets are get symbiotically associated with the mycorrhizal relationships. And majority of these plants, okay, they are gymnosperms and angiosperms. But rarely, the pteridophytic plants, they also get associated with the mycorrhizal fungi. But 95% angiosperms, and up to that, 83% dicots, and 79% monocot plants, and 100% gymnosperms are having a symbiotic association with mycorrhizal fungi. There are some exceptional cases like family Brassicaceae, Cyperaceae, and Juncaceae. They do not have a mycorrhizal association. There are several reasons for that. That they may secrete some glucosinolates or their hydrolysis products, isothiocyanates, or the Bhagyaraj in 1991, he also having the opinion that there must be some cell wall barriers and that not allowing this mycorrhizal fungi to get associated with the members of this families. But these are the exceptional cases. When 10 to 20% of the plant population, they don't have this symbiotic relationship with the plants, with the mycorrhizae. If you see the types of the mycorrhizae, there are generally two types of mycorrhiza. One is ectomycorrhizae, or it is ectotrophic fungi, which are these fungi are not penetrating the host tissues. They are present on the roots, outer surface of the root only. They forming the sheathing, hypal myceliums. Whereas the endomycorrhizae, they penetrate inside the cells and absorb the nutrients from the plant also. But in turns, they provide the nutrients or raw minerals and water to the plants. And that's why these endomycorrhizal association is very interesting. They commonly called as the arbuscular mycorrhizal fungi. Mostly they are aseptate hyphae, they are having the aseptate hyphae, and they have the most widespread distribution among the all other endospermic plants. Of course, there are some septate fungi also, like ericals and orchids. They are also having such type of the endomycorrhizae, but most common mycorrhiza are the arbuscular mycorrhizal fungi. So I'm going to focus on the endomycorrhizal fungi or vesicular arbuscular mycorrhizal fungi. They are also called as the VAM fungi or arbuscular mycorrhizal fungi. So it is very important soil microbiota. It's a component of the soil microbiota. And the vesicular arbuscular mycorrhizal fungi, they plays a very important role in absorption of the phosphorus and other macronutrients as well as the water, and they are providing to the plants. They have the obligate symbiotic association that already I explained, that this fungi, mycorrhizal fungi, arbuscular mycorrhizal fungi, they have the obligate symbiotic association with the roots of the higher plants, and they have great influence on the survival of these plants. Because without the mycorrhiza, I can say, these higher plants, they cannot be survived. So this is very important symbiote with the higher plants, and with their association only the survival of these higher plants is possible. Mycorrhizal fungi, they play a very important role in the evolution of the land plant also. Means when the plants are migrated towards the terrestrial habits and they adopted the terrestrial habit, in that case also, these mycorrhizal fungi, they played a very important role. And that's why in the evolution process of the evolution also, we can say that the mycorrhiza played a very important role. And these mycorrhizal fungi, even their hyphae are getting the fossilized in the fossil forms of the roots also. This mycorrhizal fungi or VAMs generally reproduces asexually. They produce in the endospores, or they commonly called as chlamydospores or the zygospores. Or in some cases, they may produce the sporocarp, and generally they reproduces by means of these spores only. These spores are abundantly present in the soil, and when they get associated with the higher plants, okay, they form the relationship. So these mycorrhizal fungi, when they get associated with the higher plants, they send the extramatricular myceliums, and this mycelium, they form the network in the soil, and they draw the nutrients, or they absorb the very important minerals like phosphorus, zinc, magnesium, and all, including water also, and they're providing to the plants, higher plants. So these are association with the roots of the higher plants. It enhances the growth of the plants, and ultimately all the parameters of the plants, its weights, dry weights, fresh weight, length of the plants, and finally the yield of the crops also, it get increased because of such association, and that's why we call it as a symbiotic association. Actually, there is very less information is available regarding this ectomycorrhizal fungi, and there is widespread, wide gap also, wide scope also in the research field regarding the arbuscular mycorrhizal fungi. If you see more than 3 lakhs plants, okay, it is estimated around 3 lakh plant species having this association with the mycorrhiza. So it is very difficult to isolate these plants because they are obligate symbionts. They cannot grow on the synthetic media, and that's why its taxonomical study is also very difficult, because just on the basis of the spore size, spore color, sorry, Are you clear? My audio is clear, ma'am? Madam, any problem?
Yes, no, sir, no, sir. Yes, we are audible, sir.
Okay. So it is very difficult to classify this fungi because we have only the spores. On the basis of the spores only, we can classify the fungi based on their spore size, wall size, color of the spores, okay, and the hypal attachment which is there for the spores. And on that basis, actually, the hymen in 1984 and later on it is modified by Morton Benny also in 1990. So all this vesicular arbuscular mycorrhizal fungi, AM fungi, they are included in the class Zygomycotina because they are accepted plants. And there are actually two orders. One is glomelles and second order is endogonales. In the order of glomelles, the genus like Glomus, Sclerocystis, Gygospora, Scuturospora, and Aculospora. Whereas in the endogonales, the single genus Endogone is there. So around 6 genera and 130 species are recorded over here. So there is a wide scope of the taxonomic research in mycorrhizae also. So these are the obligate biotrophs and form the large spores. Okay. Superficially, they appear like the zygospores, but they are not the zygospores. There is no any sexual fusion over there. They are asexual spores, but apparently very large in size. Their size is ranging from the 15 to 400 microns. So then that's why they're commonly called as azygospores or the chlamydospores. Now the question is arises, how to study all these spores then? Because on the basis of the spores only, we have to study the mycorrhiza. So first of all, we cannot isolate on the synthetic media. So we have to go for some techniques, okay, that I'm going to introduce you here. Some methodologies are there. And on the basis of these techniques, you can study the mycorrhiza, like isolation of the M. propagules or the M spores by bed sieving and decanting method. It is the Gardner's and Nicholson method, very simple method, and you can isolate the spores with this bed sieving and decanting method. If you want to identify, then the monographs or the manuals are available. Zinc and Prez manual is a very good manual for the identification of such fungi. And if you want to see the infection in the plant's root, then it needs the clearing and staining of the roots. So there is a Phillips and Hyman's method is there, 1917 method. It is very easy method. And with the help of this method also, you can see the colonization of the fungal hyphae in the roots. And similarly, by Moses 1980s method, that is percentage of the assessment of the root colonization, that also one can see. So this is about their different techniques or the methods that you have to follow if you want to study the mycorrhiza. I would like to share some photographs of the mycorrhizal spores. These are the, you can see this is the fungal attachment, hypal attachment is there. These are the spores. The spore wall is very distinct because all these characters, hypal attachment, color of the spores, then the wall of the spores, size of the spores, they plays very important role in the identification of this mycorrhizal spores. So all these are the glomous spores. Okay, these are again some different species of the glomus. I'm not going in the detail of this species, but this is the glomus fasciculatum. This is a glomus hoi. Okay, so these are the different spore species. Spores. This is another genus, very interesting genus, which is a Scuturospora. You can see the scutellum over here. Okay, triangular structures, scutellum. Okay, so on the basis of this, we can very identify or differentiate between the glomus and the Scuturospora. Okay, so this is the Scuturospora. These are the spores of the worm fungi. And the next genus, very interesting again, it is the Sclerocystis. It forms the sporocarp. You can see this is the large size sporocarp. It is very large. Okay, and inside which the spores are present. Okay, so you can see this is elongated spores. Okay, this is Sclerocystis pakistanensis, and this is Sclerocystis taiwanensis. So this is another genus, Sclerocystis. So if you see the specificity of this fungus, very few species of fungi are there. I already explained that there are only 6 genus, okay, and 115 species are there. But these very few species of fungi, they can associate with the large number of the plant species. I mean, they are not very specific, because see, generally pathogenic fungi are very specific to their host. But vesicular arbuscular mycorrhizal fungi or mycorrhizal fungi are not very specific to their host. Okay, so one fungal species, they may form association with the different plant species. Okay, so it is very interesting, and it is very unlike the phytopathogens. Okay, they are not very host specific, so their host range is not limited as such. Then if you want to study its structures, like morphology is very important. So if you want to see the roots, the fungus in the roots, then we have to clear the root, stain the roots, and then only you can see the different structures over here. So if you go for the macerations technique, then you can see the intercellular and intracellular hyphae of this fungi inside the roots. Okay, you can observe even if the arbuscules, tree-like branching patterns are there, which are actually they are the hosterias, and these hosterias are called as the arbuscules. And similarly, there is special structures like vesicles. There also we are observing in this case. So these are the arbuscules. You can see this is the branched structures inside the plant cell. This is enlarged structure. This is the fungal hyphae, intercellular fungal hyphae. It is penetrated inside the host tissue, and you can see here these are the branching of the fungal hyphae. Actually, the site for the nutrient exchange, because whatever this fungi is absorbing the water or the minerals from the soil, this fungi is providing to the cells of the higher plants, and in turn it takes the nutrients, ready food material from the plants. So these arbuscules are the site for the nutrient exchange. Okay, so generally they surround after they release the nutrients inside the plant cells. So sometimes the intercellular hyphae, they swells and they form the structure that is called the vesicles. Okay, so they may be at the end of, you can see this is the intercalarial structures of the vesicles, and this is the terminal one. Right at the end of the hyphae, you can see this is the vesicle, whereas these are the intercalary hyphae. You can see this is the hyphae. Again, this is a swollen structure. This hyphae is continuously growing over here. So generally, they actually store the food materials. They are rich with the lipids. Okay, so these vesicles are actually involved in the storage of the food materials. Okay, but in the latter stage, these vesicles, they perform very important role in the reproductive structures. They develop the chlamydospores, or they may develop the sporangia also. So these vesicles, later on, they develop the so if you say the structures, okay. These are the arbuscules. You can see these are the vesicles are forming, right? This is the extramatricular mycelium, and these are the later on the spores which are formed from the vesicles. So this type of the association, the plants and the mycorrhizal. There are several factors which affects the colonization of the worm fungi, its influence, and specifically the physical factors like moisture, temperature, light, the pH of the soil, nitrogen, the different minerals like phosphorus, potassium, the organic matter which is present in the soil. It also plays important role in the colonization. Similarly, the biological factors, the host plant itself, it plays a very important role in the colonization of the worm fungi. In this case, when this fungus is entered inside the host tissue, there is no any morphological changes occurs in the root. There is no any altered morphology we are observing in the roots, and that's why it is very difficult to determine which roots are infected and which is not, which are not. So that in that case we have to clean and clear and stain that roots, and then only you can go for the microscopic observation. So the cannot grow it in axenic culture. That's important. Okay, and that's why the axenic culture is not possible in this case. But in our laboratory, we have tried to develop the axenic cultures. These are the we have not got the full success in this. Partial success we got, but you can have the agar gel on the slide. You can see these are the roots of the jar plant, and where we are inoculated the mycorrhizal spores, and we are observing whether they are getting infection or not. Okay, so these are the another method. In the test tube we have developed the agar. We have taken the agar, and you can see here again that we inoculate the spores on the slide. Okay, and we observe whether there is an infection or not. So these are the different techniques we are used to see the in vitro growth of this fungal hyphae. So this is also, you can call the axenic cultures, but axenic cultures are not successful in mycorrhiza. We have to go for the traditional methods. We have to use the plants like maize or jar, and on their roots, generally we are inoculating this fungi, and then this fungi we are multiplying on it. So if you see this fungi, their interaction is very important with the higher plants. They particularly this extramatricular hyphae, they take up the inorganic nutrients from the soil, particularly phosphorus, nitrogen, okay, other micronutrients also, and water also, and they are providing to the plants. So they are actually they don't have any saprophytic abilities. They directly take the food material, ready food organic nutrients from the higher plants, and in turn they are providing all this, and that's why it is a typical symbiotic relationship. Now proceed to the effects of the fungi. So how this fungi, they plays an important role. Okay, if you take the inoculum, and if you provide this inoculum in the sterile soil also, then also you can see the effect of the mycorrhizal fungi and the growth of the plants. So you can see here, this is the most probable number method. We have inoculated the worm fungal spores, okay, in the different quantity, and you can see here the very drastic effect on the growth of the plants. You can see here the different plants, they are actually showing the different growths. So here the number of the spores we have increased, and this is the optimum number. And at this stage, you can see this pigeon pea plant, it shows very interesting growth over here. Actually, in case of the legume crops, the effect of PM fungi is very typical. It is very interesting because there are two microorganisms are involved in the legumes. One is the rhizobium, which is also symbiotically associated with the plants, and the mycorrhiza. So there is a dual symbiosis in legumes. So when two microbes like worm fungi and bacteria like rhizobium, it get associated, they shows a very effective growth, or they actually provides the nutrients, nitrogen, phosphorus, and all the nutrients to the crops. And that's why the legumes crops, they shows very drastic effects on the growth parameters, the total size of the plants, length of the crops, dry weight, fresh weight. Similarly, the yield of the crop, it also increased. And we have observed that both the symbionts, if you use in the legumes, mycorrhiza as well as the rhizobium, they plays a very important role in the legume crops. So this is the different crop, but in the monocots also, effectively, this WAM fungi plays an important role. So if you see their applications, then of course this fungi, they increases the supply of the inorganic nutrients to the plants. They are, all of you know that the phosphorus is in generally insoluble forms in the soil. So it's solubilized, it absorbs and provide nutrients to the plants. So the extramatrical hyphae, okay, which form the network in the soil, and all the micronutrients which are present in the soil, okay, they are also absorbed by the plants and providing to the roots. So it increases the ability to absorb the insoluble nutrients, specifically the phosphorus. Actually, the plant's hormones produced by this fungus, they also changes the physiological status of the roots, and the water absorption is effectively increased due to the mycorrhizal fungi. It has been reported that the mycorrhizal fungi, when get associated, it increases the tolerance of the plants to the drought conditions. Actually, there are several experiments, several research is going on this, that the WAM fungi is playing very important role in the drought. Okay, so the tolerance to the drought, high temperature, pH extremes, heavy metals, and transplantation shock. All that can be minimized by using this arbuscular mycorrhizal fungi. So in all these conditions, these fungi, they plays a very important role. If you consider the pathological point of view also, then also there are several pathogens which attacks on the root system of the plants. And it is actually observed that this mycorrhizal fungi, it increases the resistance in the resistance to the several pathogens, soil-borne pathogens. And that's why they actually provide the physical barriers between the plants and the microbes, okay? And they resist the different type of the pathogens also. So they plays very important role in increasing the resistance in the infections of the plants. They enhance the, specifically if you see, the biochemical and physiological changes generally occurs during the salt stress condition. If there is a salt stress condition, okay, then also this mycorrhizae plays a very important role because they enhance the soil water uptake. The accumulation of the proline and soluble sugars for osmotic adjustment, it is also occurs. And similarly, they enhance the chlorophyll content in the soil. Obviously, then rate of the photosynthesis also get increased. And lastly, it increases the yield of the crop plants. So in the salt stress condition also, the mycorrhizal plants, they plays a very important role. Actually, mycorrhizas are the key components of the ecosystem. You go in any ecosystem, right? So it may be grass, or it may be the herbs, shrubs, or maybe the trees. Okay, all the components of the ecosystem, they have association with the mycorrhizae. So it is very good link between the plants to maintain their habitats also. So the growth of these all plants, actually, we have also carried many experiments. So Bhagyaraj in 1995, myself and Dr. Nair, my guide, who we have conducted different type of the experiments, and it is observed that the VA mycorrhizal fungi, they are drastically improves the plant growth, and yield of the crop is also get increased. They retains and conserve the mineral nutrients also. There are several soil-borne pathogens. They are get suppressed, and it is reported by Jalali in 1979. And in the agricultural system, it is now more apparent now because of the mycorrhizal use. There are several plants, they get benefits. So there are very interesting field of the research, that is mycorrhiza. If you want to exploit this fungi, it is very essential to undertake their to understand their basic biology, ecology, their taxonomic identity. And I tried my level best to introduce you this mycorrhizal fungi, specifically the vesicular or vascular mycorrhizal fungi. And it will definitely plays very important role in development of to increase the yield of the crops. And that's why in agricultural system also, this mycorrhizal fungi will play a very important role. So I'm now set aside 10 minutes, or you can take an extra time also for the questioning. I enjoyed today being with you. And if you have any questions, are there any questions? You please ask me. I'll try on my level best. Again, thanks the organizing committee for inviting me for such a wonderful session of this webinar. Thank you. Now the topic is open for the discussion. You can ask me.
Yes, if you. Thank you, sir. Yes, anybody have any questions? Any participants? Yes, questions are welcome. I think no questions, sir, at all. Thank you very much, sir, for your excellent presentation. Very informative. It will be a great pleasure for us to listen to you again in our future events. Thank you very much, sir, once again.
Thank you, madam. Thank you.
Now, I would like to express our profound gratitude towards our President, Padma Shri Dr. Fatma Zakaria, madam, for providing such a great platform for higher education. I thank from the bottom of my heart to our beloved principal, Dr. Makhdoom Farooqi sir, for his constant encouragement and motivation. At the outset, I thank our resource person, Professor S.V. Ahire, Wadia College, Pune. We are really enlightened with your knowledge and presence. I also extend my thanks to Professor Sumaya Fatima, Head Department of Botany, for her enormous cooperation in the organization of this. Professor Vidya Pradhan, madam, for her guidance. I take this opportunity to place and record good thanks to Dr. Paithankar sir, IQAC coordinator, for the logistic support and guidance. Our special thanks towards technical committee, Professor K. Yasmin, Dr. Mohsin sir, Dr. Uma Sheikh, and Dr. Siddiqui Bushra for taking the efforts in making success. Our heartfelt thanks to all participants, colleagues, and students for their active participation. It is the warm words and a kind message. Use the end of today's webinar with the permission of me. And here the webinar is over. If you want to please save the. Thank you once again very much for the participation.